Well, hey friends, it's that time for us to start. I I've got a few questions. I should let you know that on my Patreon account, I try to do this every couple of weeks where I give people an opportunity to ask me any questions they might want ahead of time, and then I come on and try to answer them. You don't have to be on my Patreon account, although it's cheap, $1 a month. Yeah, you can help me out there. It's cheap and you can just get on there and do this sort of thing. And also, my Patreon followers are invited to join me on Zoom. I see nobody's joined us yet. Maybe they'll pop in. We'll find out. So it's for my Patreon people, but anybody can join in on YouTube and ask questions. It's all good. I'll try to answer anything that might come up. So I guess I will plunge right in. How exciting. So first question of the day is, let me fix a few things here. So my first question of the day, I was asked a very basic one, and I'm glad they did because this is, oh, hello, Joe from Patreon has joined us. Uh, this is a good question to ask because it is so basic, and this is something I always worry about in my classes is I could just barrel ahead, and what if I have overlooked some key component of the foundations for what I'm talking about? And so I always like it when people chime in and ask me questions. Uh, it's encouraged. Okay, it's encouraged in my classes. It's encouraged here. If you have any questions, feel free to chime in. And although there are such things as dumb questions, I have to tell you that, but this is not a dumb question. Okay, Joe, did you want to chime in with anything before I get started?
No, I think germ layer theory is wrong, and so I'd love to
Oh,
talk about it.
okay. I've got three questions, and that's the third one. And I will agree with you, sort of. I mean, there's a phenomena there, but it's not anything like what it was historically. You mentioned that I should look up Brian Hall. Yeah, I'm familiar with Brian Hall. I know his work, and yeah,
Neural crest as a fourth germ layer.
he's not the only one who says that.
Yeah,
There's
yeah, I agree
lots.
with him. Yeah.
Yeah, yeah, and it may be that there are others too. That anyway, we'll get to that. Let me first get to the first question, and that question was about whether viruses replicate, which I initially dismissed as because, okay, yeah, that's obvious they do. But of course, that's one of those basic things that you have to learn. But yeah, viruses do replicate. And they asked, does that mean that they mutate in infected people too? And oh boy, is that true? That is really true. Let's bring that back up. Come back, come back. Hey, Zoom, here you are. Okay, so I'm going to share a little paper with you. If I can find it in all the open windows I've got here. Yeah, I know it's in there somewhere. There we are. Yeah, so this has been known for a long time. So this, for instance, is a paper on the origin and diversity of the HIV-1 pandemic. HIV is terrible to say this. It's a great one to study this phenomenon in because HIV infections can be more or less controlled nowadays. And so you've got this long history. We've got people who've had HIV for 30 or 40 years. And all that time, the virus has been replicating inside of them. And what it means is that the infection they had at the onset of the infection could be very different from the infection that they have 30 or 40 years later, or even a year later, or 10 years later. There are papers out there that study individuals, look at individuals, and they take samples of the virus and track them and make pedigrees of how the virus is changing and diversifying. So like this paper says here, the high mutation and recombination rates during viral replication result in a great genetic variability of HIV within individuals, as well as within populations upon which evolutionary selection pressures act. So yeah, it's going on all the time. The longer you let an infection linger, the more the virus proliferates, and every time it's replicated, there's a chance of an error. So you start accumulating mutations. So that's the bad news. That's why it'd be really good to get this pandemic under control, because all those unvaccinated people where the virus is having a grand time using them as a playground and reproducing over and over again. That's those are just little petri dishes for fostering new variants to emerge. But yeah, so this paper just it's a review paper. It does an overview of all this sort of stuff. But sure, yeah, you can draw little phylogenetic trees of the different variations that are out there. So we can trace the history of the HIV in painful detail. So you can see here we got all so these are simian viruses out here. Here are a bunch of human variants. Yeah, we can trace who they came from. And then, this is a worldwide phenomenon, and different locations have different variants, different frequencies of variants. So yeah, it's real. And of course, COVID is the same thing. The COVID, your infections don't last quite as long, but still, I should have brought it up here. I found one paper that talked about tracing the COVID virus in, I think it was 4 individuals in the Netherlands, and looking how it changed over time. So yeah, this is going on. It's kind of a well-known phenomenon, and it's what you'd expect. You have a replicator, it's growing in a fruitful host environment, that is us, and it's going to be accumulating variants as it proliferates, and you might get some surprises along the way. There's all these opportunities for mutation. I would also say that natural selection can operate within an individual host as well, because a virus that fails to replicate in one of your cells is just going, that's a dead end. But the viruses that are more successful at fighting off immune attacks and thriving in your cells are the ones that will go on and eventually be spewed out when you cough on somebody and infect others. Okay, so that's a real phenomenon. The second question is also about viruses. Don't you know, I'm not a virus person. I'm not an expert on viruses at all, but I can still answer a few basic things. It says, given the choice between a vaccine that stops the spread but does nothing to reduce lethality, and a vaccine that allows spread but eliminates lethality, what would be a better strategy for us? That's a kind of peculiar question too, okay? Because the vaccines are acting on you. They're modifying your immune system, not the virus. So taking a vaccine is not going to modify the virus directly. It can do so indirectly by changing the immune system environment, and how that's going to affect, that's going to select for certain variants in the virus. But this is not a question we have to worry about, because what we want to do right now is we just want to have a vaccine that limits the spread. We want it to stop proliferating and invading more people. So that's one answer there. Another way to think of this is that we already have treatments, not vaccines, obviously. We have treatments that limit lethality. And those are working on the people who are infected. Intubating them and putting them on a respirator is a treatment to limit the lethality of the virus. So this is not an either or sort of question. We should be doing both. We should be doing what we can to keep people alive, when they're infected with the virus. And we should be doing what we can to limit the ability of the virus to spread, that's why we're supposed to wear masks everywhere, and nobody does. Not, at least not where I live. It's why you're supposed to get the vaccine, because that will limit the rate that the virus can spread through the population and thereby incidentally reduce the lethality rate. So yeah, so that's my answer to that one. Okay, Joe, it's time for you to shine, because we're going to talk about that old germ layer theory a little bit. So let me bring up, I went and found a paper on this. And of course, it's by Brian Hall. Brian Hall does everything on this. Okay, so in summary, to abbreviate everything here, so you don't have to read the whole paper, what germ layer theory says is that early in development, you set up these three definitive sources of cells for the organism. And so there's ectoderm and endoderm and mesoderm, right? In us triploblasts. If you go to something like a jellyfish, they say it's a diploblast. It's only got two layers. And who was it? Was it Lancaster who named protists as monoblasts because they just have one cell type? Okay.
Yeah.
So that's the idea, that the core idea.
Well, Lancaster also locked us into having just 3 germ layers, in my impression.
Right.
Prior to this time, Haeckel and even von Baer had talked about 4 germ layers, maybe 5 germ layers, because I was looking for the origin of how can there only be 3? And I traced it back to Lancaster, because he wrote an article where he classifies animals according to this germ layer theory, which I think is inappropriate.
Yes. Yeah. And if he'd allowed 4 germ layers, we'd have a whole nother classification of animals, which in his scheme, which wouldn't hold up very well at all. So anyway, yeah, so this was a 19th century idea that we can describe organisms as combinations and interrelationships between these 3 germ layers. And that's what an animal is. And the initial idea was that these were inviolate, that you would have these germ layers set up and they're predetermined. So ectoderm is going to make skin and nervous system. Mesoderm is going to make muscle and connective tissue and blood and things like this. And endoderm is going to make the lining of your gut and some of your glands. And that's all you can do with that. And that's definitely not true. More modern versions of germ layer theory accommodate the idea that you can have change in the patterns of the descendants of these cells. And I would say that probably the definitive change was with Schmemann and Mangold, who showed that experimentally you can pluck out these different layers and you put them in different environments, and presto, they transform into the tissues of their new host environment. And also that this is also where we get the idea that what's going on, and the reason we have these 3 layers is not because they define blocks of tissue, but because they represent different developmental trajectories, and then allows us to get more combinations of tissues by interactions with each other. So, for example, the key thing, the big thing that Schmemann and Mangold and all those people in the 20s and 30s and 40s were looking at was induction, right? So you transplant a bit of dorsal lip, this little piece of the embryo where involution, where cells are rolling into the interior, to anywhere. And what it does is it instructs the tissues around them to become the axis of an animal. So obviously, the fates of cells are not fixed early on.
Right.
Yeah, so they can change. Then the thing that Brian Hall is focusing on is mesenchyme and the neural crest. So there's this tissue called mesenchyme. It's, as you can guess from the name, it's supposed to be mesodermal. And what mesenchyme is a loose aggregate of cells that are migratory, or in some cases just can expand, and that are going to then differentiate into things like muscle. Okay. But there's a certain class of mesenchyme that's especially special, and that's neural crest. So neural crest arises from ectoderm. Yeah, so when the embryo folds up to make a neural tube, there's this little bit of the ectoderm at the top of the neural tube that just starts migrating out, wandering around in a little matrix of glycosaminoglycans and cool things like that. And it's mesenchyme, but it's of ectodermal origin. So there's another heresy for you.
Okay, so one of the biggest heresies I have a problem with is, how did muscle evolve? How do we not have muscle before mesoderm appears? I can see a very progressive evolution of muscle from diploblasts into triploblasts with these epitheliomuscular cells. Two
Right.
layers of them, just like we have two layers of them.
Mhm.
So to me, yeah, the mesoderm breaks down because how do you, we have ectodermal muscle, right? The erector pili muscles have to come from
Oh,
ectoderm.
right. Yeah. So that's the thing is, it's not a hard and fast distinction, because we can get these different tissues arising from different areas of the body. If you just have the right signal, the right inductive signal, you can get transformation into other tissues. So yeah, that's an issue there. How did muscle evolve? That's an interesting question. But
do you mind if I throw my idea at you?
Yes, please.
Okay. So in the Cnidarians, they're two-layered animals, essentially. We call them diploblasts. And they are essentially just two layers, like a water balloon that's sort of squished up into itself, a membrane on either side. And those epithelia, they're largely epithelial, but at the base of them, they have this muscular extensions that go out. On the inside of that tube of their body, it's a circular layer of muscles. And on the outside of the tube of that body, it's this longitudinal layer of muscles. Okay. That's exactly the layering of muscles we see in our gut. We've got this circular layer inside a longitudinal layer. So,
Right.
to me, yeah, you don't need mesoderm to explain muscle. Muscle surely must have evolved from these epitheliomuscular cells.
Yeah, well, and also Cnidarians, we have this other tissue. There's this thing called mesoglia. Yeah, there's mesoglia. It's, for some reason, it's not counted as an embryonic layer, presumably because you don't see any kind of coherent organization and formation of this massive tissue. Instead, you form ectoderm and you form endoderm, and what's in between them is not vacuum. It's full of extracellular goo and this thing called mesoglia. And yeah, you could imagine it evolved from those as well. That the development and evolution are much more fluid, and you can't just say, well, it was set up early and that is determined and it's never going to change, because it clearly does change. And Hall is pointing this out, that neural crest is a great example of something that diminishes that boundary. It really makes it much more interesting and much more complex, because they're ectodermal, but they act like mesoderm. They look like mesoderm. So why are we continuing with this distinction? Well, it's more historical and kind of histological, that if you look at an embryo, you can see these regions, right?
Well, but I think we should look at animal evolution in terms of tubes. We're mostly made up of tubes. I think germ layers was a bad start to looking at animal evolution, because he was looking at chicken embryos that he cut off the top of the enormous yolk, right? So it looks like 3 layers because the rest of the tube is gone. But there's this famous figure with a frog embryo that looks just like tubes, and a chicken embryo, you cut out the yolk and you pull the edges together. They're tubes again.
Yeah, I mean, I would kind of disagree that it was a bad idea. I think it was an initial idea that has to be revised. And, I actually use it as an example in my introductory class. So, we're talking about theories, and I'm saying, what is a good theory? And if it's a bad theory, do we throw it out? And, I mentioned, for instance, Haeckel's theory, the biogenetic law. I say that's one we've just thrown it out entirely because it doesn't work at all. And then we have germ theory. Sure, we'll stick with that one. We'll also stick with cell theory. Seems to hold up fairly well.
We have to have CLOM theory to explain why we have the body cavity. Yeah, there's no really,
yeah, but germ layer theory I throw out as an example of something that's kind of in between, where, okay, we can see the phenomenon. It's still an observable fact in many ways, but it's lost a lot of its utility because we do think of cells as being much more fluid than how they were represented in the 19th century. Isn't it good to know that we can progress beyond the 19th century? Yeah,
it is.
And Hall mentions also this thing. So this is a little diagram from the paper. So he's making the point that we've got this variation even within the tissues. Mm-hmm. So this is ectoderm. And he's saying, yeah, we can sort of categorize what layers of the ectoderm will become by initial variation in this one signaling molecule, bone morphogenetic protein, or BMP. So we got this one. If it's at a high concentration, those cells are going to turn into epidermal cells. If it's at a really low concentration, we're going to make neurons. If it's at a middling concentration, we're going to make neural crest. And I think this is kind of an important concept right there, is that we can, it's not simply a trinary or binary or however you want to divide it. We've got gradations that generate distinct differences in the fate of the cells. So we can use a gradient to define identities in tissues. And
interestingly, the gut tube and the central nervous system tube both use BMP and Saunders-Hedgehog gradients to lay themselves out in that direction.
Right. Yeah, you can see that there's this interesting BMP and Sonic form a kind of opposition, and you can actually, if you manipulate SHH and drop it down, you get an expansion of BMP and vice versa, and you can get transformation of tissues in that way. But again, what you see here is this is a pathway that leads to neural crest, and it's got distinct genes that are switched on here. And if you look in muscle cells, you'll also see, for instance, snail is going to get expressed in muscle. So this is a transformation that makes it look like classic mesoderm, and it's just driven by the signaling molecules that are present. So yeah, that's the thing. It's, I wouldn't throw out germ layer theory because it's useful for describing the organization in early development. Okay, we can do that.
The only reason I'd like to throw it, and I don't want to take all your time, and if you have other questions and stuff, please you get to those. Okay. The only reason I'm willing to throw out germ layer theory is because I believe I have something better.
Something better.
I would love you to destroy it if possible, because I've seriously spent a decade looking into these things.
Okay.
Here's just a quick summary. Okay. So germ layer theory says that me and a hydra are basically the same, except the inside skin of the hydra, the inside skin of the hydra gut is the same as the inside skin of my gut. And the outside skin of the hydra is the same as my outside skin. And the difference is mesoderm filled everything else in, right?
Yeah.
I think it's far simpler to think that a hydra, which is essentially just a long tube, wrapped around itself and then attached at the other end. Now you've got a tube inside a tube. So you've already explained CLOM theory. You don't need that anymore. And you need to break down germ layer theory a little bit for this to work, but I think it works far better. I'd love to get into a more detailed conversation.
Okay.
About this with
you. But then again, you're gonna run into conflicts. Yeah. People will disagree with you because it's really there are complex phenomena going on there. Mm-hmm. So if you're going from your initial tube to a tube dual tube system with a tube within a tube.
Yeah.
There's a couple of easy ways you can do that. One is you just described, you can have expansion and migration of the tubes to wrap around.
So the gut is the inner tube of life, is the whole issue of science. So what's the outer tube of life? The body.
Yeah, so that's why, but again, when you look in development, there are different organisms that do that in a different way. Some of them don't have the clear-cut migration of one layer into two layers, and one on top of the other.
Oh yeah, there's all sorts of crazy ways. Embryology may have taken shortcuts or changed.
Yeah,
things. Absolutely.
There's also delamination, where you got a layer with made up of a single layer of cells, and the cells divide and they separate out transversely, and you right away get two layers there, and it's not clear exactly which one is the primitive method that occurred.
Let me give you the thing that really clinched it for me. This is that the fact that on the inside in our gut tube, on the innermost part of it is the dermis, the gastrodermis, and then the circular muscles, and then the longitudinal muscles, and then the peritoneum. In the body tube, it's the exact inversion of that. On the inside most is the peritoneum. Then the deepest muscles of our body are longitudinal, then the circular muscles. Then we have our dermis. The epidermis is on the outside. It's completely radially inverted, which would be immediately explained by a tube inverting around itself.
Oh, yeah, but it's also explained by the coelom, right? Because
how do you pronounce it? I always say coelom.
Coelom is
coelom.
fine.
I've been saying it wrong forever. Okay.
I've heard different
coelom.
People pronounce it different ways.
Okay.
Don't consider me authoritative. Because the other way you can do that is you've got a mass of mesoderm in the middle, and then it opens up with a vacuole of space in the middle, and they push out to be adjacent to the ectoderm and adjacent to the endoderm, and then those epithelial layers are inductive, and that's what leads to the formation of the inversion of the layers in the mesoderm. So you'll have to.
Okay, I can see your point. Yes. Okay, that would also explain it. I'd like my idea better. And
but you have to do the experiments and show us all why it's better.
Well, I would love it if you could come up with an experiment that might test an idea like this.
Oh, well, the one way I would think about doing it is if you isolate for instance, if we're testing the coelom theory, and if you could separate out the mesoderm, take the inner layer and the outer layer and culture them separately, and then place, for instance, epithelial stuff adjacent to them, what that would predict is that the formation is going to be, in the direction imposed by the epithelial layer. So yeah, we could do that. It sounds difficult because those early mesodermal layers are so gooey.
Yeah.
Yeah.
And I'm a computational neuroscientist, so I don't do any experiments of any kind anymore. Well, I mean, computer experiments, but
right. Yes, and then something might be more up your alley is we should be doing more comparative analysis of the molecules involved here. So, for instance, Brian Hall's paper mentions that when they look at the genes involved in differentiation of the endoderm, those seem to be older.
Yes. Oh, yeah.
Than ectodermal genes.
Oh, yeah. There's this great paper that was in Nature a few years ago by out of Israel, spatiotemporal transcriptomics of the endoderm germ layer, something. And it's fantastic. Yeah, the endoderm is clearly older, activates earlier.
Yeah. Yes. So then, but that does that necessarily fit better with your theory than this?
Here's a problem. So this is something I haven't made quite clear yet, is that this requires a breakdown of germ layer theory. Okay, so part of what germ layer theory calls mesoderm, I'm saying is always part of the tube. So the splanchnic mesoderm wrapped around the gut was always part of that gut tube. It's not a separate mesoderm. And the somatic mesoderm of the body tube was always part of that tube. It didn't come with mesoderm. It is the evolution of those epithelial muscular layers.
Okay. Well, and there I'm just thinking there have been a lot of studies done, with lineage tracers, for instance, on where those layers come from, and they do seem to have a coherent origin in mesoderm, in primordial mesoderm, in the early embryo.
Well, sure.
Yeah. And again, I'm not sure how that would distinguish these two ideas.
Well, no, so yeah, I tend to discount the embryological stuff because so many shortcuts happen, so many breakthroughs happen embryologically that I think it's hard to see the original physical
Oh yeah,
evolution in the embryology.
yes. So
Often.
yeah, that's always a continual problem, and Brian Hall is very aware of this. That the functions we see in modern organisms aren't necessarily the same functions they had primitively in evolution.
Right.
So how do you sort those out? I'm kind of the mind that the proper approach is not to get hung up on any particular theory about the origin of these, but just ask what molecules are being expressed, how are they affecting each other, and then do comparative work, right? That's where we're going to get the answer is we should be looking at these different molecules in cnidarians and arthropods and mollusks and vertebrates and asking how their mesodermal origins differ. Snail seems to be kind of everywhere, so I don't know how that would separate it out. Fox D3, I've seen some work.
So, yeah, and the molecular stuff is very interesting. So yeah, I mean, the crux of this idea is essentially that tubes become able to pull in and wrap around each other and do different things. And obviously you have to have some sort of molecular mechanism causing that to happen. Snail is probably involved. So yeah, from the original gastrulation, right? I forget those molecules, but similar ones are used then in neurulation. Anywhere you're pulling a tube in, I'm guessing you're going to find similar molecules causing it to happen.
Yes, and so that's what I'm saying is I am not going to get married to your tube theory because I think there's going to be some other perspective that comes along and is much more informative. We'll see.
I've got a video presentation on YouTube I would love to share with you that I gave at my university here.
Sure, yeah, go ahead.
If you would watch it, I would love to have your critiques. Tear it apart. I want it to be good.
Yeah, send it in. You can leave it in
I can share it with the group here and anybody can watch it.
Sure, that's what I was saying. Just put it in the chat here.
Perfect. I'll do that now.
Yeah.
Oh, this is great. Thank you for
sure. Yeah.
Talking about all this. I love it.
Okay, we do have some comments here. Let me take a quick look at these. So Joey asks, are you on the side of viruses being alive or not? I've heard good arguments on both sides.
Life or death is a spectrum. Alive, not alive.
Yeah. My answer to that would be that viruses are chemistry and so are we. So where you draw the boundary is kind of arbitrary. I'm not going to get into it. Let's see, Hemi Scott asks, isn't there a variation in a human cell receptor that influences, decreases susceptibility to HIV? Sure, there's CCR5 delta that affect delta 32, which affects the ability of the virus to ingress into cells. But in reply to the previous question, yeah, we can't select for that. Yeah, that's something, that's a human property and it affects all of this. But what we'd have to do then is, yes, it has a higher frequency in Scandinavians. So let's just wipe out everyone else and just have Scandinavians take over the world. They'll be more resistant to HIV. Oh, yeah, that's a bad, yeah, anything where you're tinkering with the biology of humans is you're on a slippery slope, a pretty strong slippery slope. Let's see, Scott also says BK Howell, love him. Yeah, he's a really interesting guy to read. I read a lot of his papers. I jump up when I see them.
Yeah, I love Brian Hall also. He's a really good writer and interesting stuff.
Yep, yeah. Let's see, we have something from somebody called Raw Nakedness. PZ Myers, you beautiful specimen. Well, I'll agree with that, but I suspect that you're trying to get me to go to a porn site. So I'll check later and clean you up. I'm sorry. Okay. Scott also makes a point, Gould's historical origin versus current utility. Yeah, that's always the conflict. Yeah, this is what killed Haeckel's theory, right? Is because he didn't make that nice, tidy distinction that we've got. Yeah, there he is. That there's maybe a historical origin, but you're looking at current utility and then you're trying to infer historical origins from it. That's a risky business. You don't want to do that. Okay, and Joe, did you put up, let me see.
Quick question. You keep saying that Haeckel's one of his theories has been destroyed. I've read a lot of Haeckel. I really love him. He gets a bad name for the figures. But other than that, what problems have there been?
Yeah, I would about Haeckel, I would say I actually like Haeckel a lot. He was a good guy, he was kind of bombastic and all this sort of stuff, but very German. But
highly talented, an amazing artist and scientist.
Oh yeah. And that it's true that his fall from grace was driven by a few figures that he clearly copied from another source, his own source, but he recontextualized them and that distorted them. So
yeah, I agree that was bbad, but you shouldn't judge this amazing man for one little thing like that.
But what killed his theory is not that, it's that it was von Baer who destroyed his theory before he was born. That that Haeckel's theory implied that there is a recapitulation of evolutionary ancestry in development, and there isn't. And it's because of what Scott just mentioned, current utility. It's constantly getting broken down and revised to fill the needs of the organism. So we've got this constant drift away from whatever the origin was.
But I mean, ultimately and literally, ontogeny is, or phylogeny is ontogeny, right? I mean,
no, I would disagree.
Okay, in that the cycle of birth and what led to us is each embryological step is phylogeny.
Except it's not the kind of phylogeny that Haeckel was proposing.
Yeah,
it just doesn't work that way. That what von Baer pointed out is that, for instance, all these different embryos that resemble each other, they are not resembling the adult form of the species. They're resembling the embryonic form. So what we've got is a general vertebrate embryonic form, which is kind of worm-like with somites and fringular arches and all that kind of stuff. And every species, every lineage has modified that. So sure, if you go rolling back the tape and looking early on, you'll see that it all goes back to this ancestral similarity, but each of us has evolved in different ways, and that's not captured.
Okay.
Yeah. So it just, the whole theory just kind of falls apart at that point. That at the time, the idea was, hey, all we got to do is go out to this nice marine station for a summer vacation and collect embryos, and we draw the embryos, and we have done evolution.
Exactly.
We've mapped out it. No, you have
collected all the stamps.
Yes. So it doesn't work that way. Yeah. So no, I'm sorry. As much as I admire Haeckel, he got so infatuated with the elegance of his theory that he forgot that it had to be demonstrated. And it just didn't work.
Sometimes they're just so elegant though.
Yes, and it's really a pretty idea. It just doesn't work. Yeah. Let's see. I'll get to you, Adam, in just a moment. Just so Scott says, Mayer and others agreed Haeckel didn't mean adult characters are repeated, and also perhaps some character states and not. Well, first of all, don't use Meyer as a source. Meyer is not trustworthy on any of this. Yeah, he was probably great on bird taxonomy, but when he gets in development, he's embarrassing. Um, that so Scott is saying that he didn't argue that adult characters are repeated. Ooh, yeah, he waffled a lot in his words, and you can go back and reinterpret them to say, oh yeah, well maybe, but that just tells you that his writing wasn't clear on this.
Well, yeah, my impression of the situation is that, yeah, today there's this like strict recapitulation theory, which is that the actual adult stages of ancestors are present in the embryo. And they ascribe that to Haeckel. I've read a lot of Haeckel, and I've never heard Haeckel subscribe to a strict recapitulation theory.
Right. And that's part of the problem. He was so waffly on this stuff. And you go back and yeah, and he did talk about terminal addition of genetic properties, right? But his theory also said that you could, delete wholesale sections of your evolutionary record from your ontogeny, which you can then say, well, then, yeah, maybe we can sort of assemble it if we just, and then you can also he says you can insert things. And at that point, why bother with the theory? Yeah, why go through this nonsense of recapitulation?
Special cases.
Yeah, if there's so many special cases that you can completely revamp the theory to be something completely different. So yeah, we don't endorse recapitulation anymore. It just doesn't work. Okay, so Adam asks an interesting question. Could I ask if the disease in the medical thriller The End of Men, which I have not seen, came out in April this year, makes biological sense as the medical characters in the book explain it? I suspect hell no. Okay, I don't know what happened there. So that what happens is 91% of men and no women die from disease. The explanation is that 9% of the men who survive have a particular kind of X chromosome. Women all fine as they have 2. Wouldn't 80% of the women also die? When I started reading this, I was thinking, yeah, there's a way this could happen. You can imagine a genetic disorder or for instance, a parasite that targets particular molecules and is going to differentially affect males versus females. And we already have examples of bacteria in fruit flies, like Wallachia, that selectively affect different sexes. So I can imagine that. But their explanation that 9% of men who survive have a particular kind of X chromosome. Women are fine because they also, so the proportions don't add up. You would expect, it's like color blindness, right? Much higher frequency of color blindness in males than females. So imagine a lethal form of color blindness where it would kill many more men than women. We could imagine that. We could turn that into a thriller of some sort. But the proportions are kind of outrageous. I just don't see those numbers adding up. But as a thriller, as a story, yeah, I could imagine a disease that selectively affects men with a higher frequency than women, and maybe produces these pretty profound asymmetries in expression. But yeah, I'd have to read the book to see. I kind of expect from the description here, I would say that it's greatly exaggerated. But it would be an interesting situation, wouldn't it? If a horrible situation from my male perspective, if 90% of the men all died and none of the women were affected. Ooh, yeah. It sets up some interesting possibilities. And I could imagine scenarios where that would happen. Yeah. I mean, basically, that's what some of these maternal effect mutations do, is they selectively kill off males, but it's all done embryonically. but you could imagine some maternal effect trait, for instance, that kills males at puberty. Could happen. It wouldn't happen all at once. You wouldn't suddenly see everybody expressing this particular trait, but it's an interesting idea. Okay, if 91, so Adam expands on this. So male versus female disease, okay, yes. But if 91% of X chromosomes are the bad kind, wouldn't 80% of the women have 2 bad copies? Yeah, that's true. Yeah, so that's what would happen. So we're saying that 9% of the X chromosomes are good. So we'd expect 9 times 9, we'd expect 0.18 of the females to have it. Yeah, you could do math like that with this. It would work. Yeah, you would, but you're right. If it's just simple, simple allelic trait, sure, you should have seen more females dying. Okay, Joe's still there. Can't find Joe Graham's lecture. Maybe you can email it to me and
I just posted a link again. It's there now.
Oh, it is good. Okay.
Yeah, for some reason it didn't show up earlier.
Yeah. Okay, good. So I'll take a look at that. Let's see. Berger, you say Lev Smolin's idea that black holes may create new universes is also a beautiful theory, but it appears to be wrong. Sure. Yeah. Don't ask me about astronomy. Ooh, it was bad enough I had to answer questions about viruses. Anyway. Yeah, so there's all kinds of theories that get thrown out, and the question is, do they survive the test of time? I don't think Haeckel's theory did. Germ layer theory, it has survived the test of time, but only by radically changing in its interpretation. So yeah, we'll see.
I see somebody mentioned something about Von Baer and he suffers from the hourglass. I'm vaguely familiar with the hourglass of evolution, but what is that? Could you talk on that?
Yeah, so the hourglass refers to the fact that if you look at embryos over time and you compare lots of embryos, what you find is huge varieties early in development. For example, the presence of large yolk sacs. So early embryos are radically different from one another. The phylogenetic stage, the pharyngula stage, is later, much later. So they all kind of initially converge on this one form, the pharyngula in vertebrates, and then they all diverge later. So that's the hourglass. Okay. So yeah, this is now the typical explanation. It's a von Baerian explanation for why we see so many similarities between embryos at one early particular developmental time, that pharyngula stage. And it's because what you're seeing after that stage is von Baerian trajectories. They're all diverging from one another. So they're all separating to produce the different adult forms. Von Baer didn't really talk about the earlier stages. A good source for that kind of stuff is William Ballard, who did a lot of work on gastrulas and things like that, looking at comparative work on them and documenting all these vast differences that you find in the very early embryo. Compare a chicken egg to a human egg. They're really different. And that affects the early trajectories. But they still all converge on this phylogenetic stage.
Thank you. Thank you very much.
Yeah, okay, so again, we get a comment here that your link is not showing. Oh, let me see, where is it?
I posted it on Patreon and in this chat, but I guess it doesn't show up in the chat.
Oh, okay. Well, I can pull it off of Patreon and I will post it at the bottom of this video. Great. Once we're done.
Perfect. Thank you.
Yeah. Yeah, Nestle points out, I have to give you mod control. Oh, I don't know if I can part with that much power. We'll see. Maybe in the future we'll see.
We just met. Yeah.
Yeah. Yeah, and you get this crazy idea to throw out germ layers. Oh man, how can I trust you ever again? No, it's good. I kind of agree with you in some ways. Okay, Berger points at gender differences. The Amazon molly seems to get along without males, but they occasionally mate with closely related species, explaining why their genomes remain healthy. Oh yeah. So I had a colleague who has since moved on to Indiana, but he was studying salamanders, this really interesting species of salamanders. They're all females, and they reproduce parthenogenetically, but they have to encounter, this, what is it called? The little sperm plug that they have to go through kind of a pseudo courtship mating ritual to trigger everything happening. So they're still kind of dependent on other males of other species to get there. And also what's really interesting about them is that they're parthenogenetic, but sometimes because they're encountering the sperm from other species, they will fertilize with that. And so you'll get the introduction of genes from these other species into their line. And what he was studying was that this actually is kind of advantageous. So parthenogenesis is great in the short run. You would think this is how evolution ought to work. After all, I am perfect. I had to just clone myself every time I reproduce and make more little me's to spread out over the world. That falls apart in the long term, though, because environments change, and maybe a little me running around in 100 years would be doomed to destruction, just a total failure.
And it was my impression that the immune system is one of the big reasons for sexual reproduction.
Right. That's a big one, is because the environment changes, and one of the key components of the environment that changes fairly rapidly is things like viruses and diseases and all that. So you want to keep changing to be a moving target so that the viruses don't home in on you. Anyway, these salamanders reproduce parthenogenetically, but they kind of get the best of both worlds, because every once in a while they will take in genes from this other species and mix that with their own genomes. They get a little bit of change there. And in particular, what they seem to do is take in genes that are, and keep genes that are most advantageous for the environment the donor species was living in. So if you're invading a new environment, and you're adapted to another environment.
How would they know these are good genes to keep?
Oh, they don't know. This is selection.
Got it. Okay.
So you go in and you snatch genes from the resident population, and you make lots of progeny, and then the ones that have acquired the useful genes survive and persist into future generations. So it's kind of a clever way to do this stuff. Usually, we say parthenogenetic species are kind of neat, but they're not going to be around for long. Something is going to change. They're not going to be able to cope, and they're going to die. And the most common thing that will change is the presence of other diseases. Oh, let's see. If I say show that there with any luck, Joe's link is now available. Let's see. Nestle points, I've seen that species that reproduce fast have many mating types, while those that do it infrequently have two. Yes, if you look at yeast, how many mating types do yeast have? I have no idea. I'm not a yeast expert, but they have lots of mating types, and they also reproduce rapidly. And the whole point is, when you have a mating type, you are saying, I cannot reproduce with this fraction of the population. So with two mating types, I cannot reproduce with half the population. Okay. If we had a couple more mating types, there's a science fiction story for you. If we had more than that, For instance, maybe I'd only be able to reproduce with 7/8 of the population, and 1/8 wouldn't work because we're the same incompatible mating types. So yeah, yeast, if you're rapidly reproducing, you get large populations, you may want to have more mating types because you can still prevent inbreeding. You won't be breeding with individuals with the same mating type, but you still make yourself available to a larger proportion of the population. Okay. Let's see, are we winding down here? Are we getting to a good spot? Oh yeah, the other thing I needed to do, I got one more thing to do before we depart is, let's see. I said that today would be the day I kill Facebook for myself, just for myself. So let me stop this, and I'm going to switch to my Facebook account, and we'll go through the steps and see if I can make it work. Let's see, where is it? Oh, come on. Oh, there, is that it? Yeah, I think that's it. Again, I should clean up my windows before. All right, does everybody see my Facebook account there? You don't need to see most of it, but there it is. Here's what I'm going to follow these instructions. So the instructions say, to permanently delete your account from your main profile, click the little triangle right there and select Settings and Privacy. Then click Settings. They're not making this easy. Click your Facebook information in the left column. There it is. Okay, I'm finding it. It says click deactivation and deletion. Hey, that's it.
Did you download all your stuff before you deleted? Do you want to?
I don't want to. I don't care.
No worries then.
Yeah. I mean, really, I have used Facebook just casually for chatting with people, and I didn't put anything valuable on there.
Good for
okay,
you.
there's the window. Delete account. This is permanent. So you're all going to see this. I'm literally destroying my Facebook account. Continue to account deletion. Let's hope that's it. Oh, come on. Oh, I would get rid of Messenger. I get a lot of chat from. Oh, okay. Well, first of all, I'm not going to worry about any of this stuff. So I'm not going to download the information. I am the sole admin of these sorts of things, so I guess they're going to go too. Let's just say goodbye. I'm fine with saying goodbye to all of those. Oh, please, and oh no. What is my password? What if I can't remember my password? Oh,
that's their final step.
oh wait, I remembered that. Let's see if that works. Yes. Okay, so if you are a Facebook friend with me, this is nothing personal. I'm not trying to get rid of you. And there we go. It's gone. That wasn't so bad. Okay, I've also, unfortunately,
good for you. I'm tempted to do the same, but I'm not quite ready.
Yeah, well, I've kind of noodled around about doing this for a long time.
Okay.
And it's my friends and family that keep pulling me back in because they've got stuff they say, and that's still the case. It's losing that. That's kind of unfortunate, but yeah, Facebook is evil. I'm sorry. It's just there's no denying it anymore. It's just an evil, corrupt company. It's
or
also
perhaps amoral would be the better term.
Yeah, okay. That if you're being too nice to Mark Zuckerberg. Yeah, I think he's actively evil because I mean, look at where Facebook started as a scheme for rating hot girls on campus.
That's true.
Right. And it's then he made it more commercial, but it's been getting progressively more nastily commercial. So and and I'm really not interested in providing money for them. So they're gone. Yeah. I still have Twitter. I still have Instagram, which I understand is a Facebook-owned company. So I'm still accessible. But Facebook is just not the right way to do it anymore, I think. I'll also mention, every time I go into Facebook, I am inundated with ads and clickbait. I will see a post from a friend, and then below that will be 2 or 3 clickbaity things. I'm supposed to, sponsored content or whatever. And I just want all that to go away. And so now it's gone forever. Good. Now the rest of you can, no, you don't have to. This is my personal choice, and whatever your choice is, you should follow it. But I'm not going to worry about it. So it's gone, gone, gone. Yeah. Well, that was a good way to end this particular session. So thanks everyone for stopping by. Thanks, Joe, for throwing ideas at us.
Yeah, thank you so much, PZ.
Sure. And let's one last question, if it's a quick one. Go ahead, Nestle. Fire it off, but it better be quick.
Oh, I see. She asked it above there.
Oh, it's above.
About the body axis of jellyfish. Is that homologous to our anterior posterior or top bottom axis? I've asked PZ about before, but I wonder if Joe has remarks on it.
Yeah, go ahead, Joe. Fire away.
Okay, so you want to know about the body axis of a jellyfish. Yeah. So I use a water balloon model of early evolution to help me. So yeah, a jellyfish would just be like a water balloon, like pushed up inside to make concave. And then you can pull that out. A hydra, which would be anterior posterior as it sort of extends itself into two. I think it's going to be anterior posterior because it's like the gut through axis.
Right. But that's where the problem arises, because then you look at other organisms and you've somewhere got to get dorsal ventral in there. So yeah, coelenterots and cnidarians, we got oral, aboral, that axis set up. But I don't think that corresponds very well to anterior posterior.
Yeah.
We've got to add another axis of information in evolution for that to work. And yeah, so is that homologous? I don't.
Exactly. They've changed so much since then that homology.
Okay, but with that, I will let us all go. Thanks for stopping by, everybody. It's been fun. I'll do it again in about 2 weeks. So think of questions. You can email them. You can send them to Patreon if you're on Patreon, and we'll get to them then. Okay.
Thanks again, PZ. Bye-bye.
Yep. Bye.